I really owe a lot to these gentlemen who allowed this newcomer to be the expert. Even though the growers wanted answers from them, they said, nope, Sue is the breeder now. And that was wonderful. They welcomed me and so many collaborators. So there's a lot more to acknowledge. My technical staff, Kevin Maloney and David Terry. Kevin was a Cornell alum. Colleagues past and present at many institutions. Our field research, we wouldn't be anything without the guys that set up trellis, mow, spray. Students and summer scholars, Cornell Cooperative Extension, and then crunch time apple growers who I'll explain later. And those of you in the audience, because whatever field you're in, your research has benefited the next generation. And we may not always say it, but we're very appreciative of your efforts. Now, I wanted to do family genetics because one, I'm dedicating a book that I wrote on creating an apple to my mom, who was a green thumb extraordinaire. And she was also a woman that started in the workforce working nights at the post office. I found out where my son's insomnia came from. That's how she dealt with hers. My father was a pigeon breeder and worked at a nuclear power plant. And so from that great Polish melting pot of Kotowski by Balzakowski, you can see that we had, I don't know if there's a pointer here, and if there's not. So the lowest picture is my nephew, my son, my husband, and my brother Bill. And you can see that the genetic diversity is not so distinct. They all look like they could be part of the same family. My sisters at a wedding, that's me, my daughter Lauren behind. Lauren's a Cornell grad, my sister Lynn and Carol. So the reason I do this is not just because I'm proud of my family. My daughter did a family tree and she put my husband's family on the ground, which we refer to as drops. I haven't told them that. But the fact that as a breeder, we're taking, just like you having children, we take two parents and we want the offspring to be better than the two parents. And we also want it to be distinctive. But as you can see in a family, getting it distinctive is tough because people would say, oh, that's a Kotowski girl because I look like my sister and my sister looked like her sister. And so then this is my family, my son Michael, me, my husband, and my daughter Lauren. And again, you can see that the old adage, an apple doesn't fall far from the tree, holds. So for crosses, I tried to explain to growers that genetic modification is what happens in everything. So Snapdragon is a hybrid, genetically modified from its two parents. If you planted a seed of that apple, it would be a unique hybrid. And so I was trying to explain to a grower and I said, well, let's say you and your wife, who's beautiful, have a child and everything's fine, but she gets some of your genes. And the growers are like, Dr. Brown just dissed you. And so, there's genes on good sides and bad sides, but they're not GMOs. And so the distinctiveness is tough. If I cross Gala by Honeycrisp and I get stuff that just looks like Honeycrisp, because we mark it by name, I have to, I throw that out. So what I love about working with apple, as opposed to people that work with wheat or corn, is when you work with wheat and corn, you want it to be uniform. And apples just scream diversity. And that to me was fascinating. So every seed is a unique hybrid because apples are outcrossers, which mean they need pollen from another source. The bee brings it, or in the case of an apple breeder, I bring it. And that's why you have to plant several trees in your yard to allow that cross-pollination. So this is the process of hybridization. So we collect pollen and also remove the petals at this popcorn stage that looks like a little ball. And so we will collect flowers and then screen them out. And fortunately, kids and graduate students are wonderful for screening out pollen. Even my technician's wife says with a glass of wine, it's not such a bad job. And then we do something which is the indelicate term of emasculation. We remove the petals and the anthers. That allows no bees to come and make contact. And then we put pollen from another tree on those emasculated flowers. Now, my husband has a good sense of humor, but we met at a sports bar, and he goes, "You look exhausted." And I said, "I've been emasculating all day." And he said, "You really shouldn't say that in public." Now, I still do. So our goal is to produce superior apples. We need it to be consistent, because if you get a bad apple, that you're likely to not buy an apple again, to go to a banana or a clementine. We want it to be crisp. That's the primary quality that people are most interested in, even over flavor. And I say we want to be the chocolate of fruits, because who doesn't like chocolate in this audience? I thought so. So when I first gave my daughter chocolate, she tried it, and I waited quite a while, and she went, "Oh, mommy." And my husband said, "The gene has been turned on." So we would like apples to be a treat, and they should be. Better nutrition, unique flavor profiles, but better requires very complex traits to be modified. And if you've taken genetic classes or heard about Mendel and his peas, there's a lot of single genes, but in reality, everything's really complex. And the more we know, the more we have to know. And so that it's going to be the next generation. So we need new varieties for tomorrow, but new information too. So I'm breeding for quality. I'm really fortunate, and I hope you'll see, I get to play around with a lot of stuff, and people will say, "I didn't know you bred crab apples." And I just go, but in reality, they're going to be patentable and support the program. I have always been passionate about what I do, and the toddler stage when my kids were little was really annoying, but I'm stuck at that stage. I'm always asking why. Why does this happen? What does this happen? How does this happen? And that's a valuable skill that I hope to encourage in the next generation. And I love reading the literature, and I've got my students to do that. Climate change is going to be something that we have to work on. But the difficulty is, how do you breed for unpredictability when you get drought one year followed by a flood the next? So some fast facts, because there's 54,000 genes in apple. So it's been a lot of apple varieties have been sequenced. In comparison, that's more than humans. And it's because there's areas of duplication. When the apple evolved from China and Kazakhstan and other areas, it was actually a glacial refuge. So they were able to grow and evolve. And so they've been around forever and did a lot of changes, but there were duplications. There's 17 chromosomes. As I said, many areas of genetic duplication. And then something I'll delve into a little bit more about areas of transposable elements or jumping genes that contribute to the diversity. We have 25 to 34 different malus species, and sadly, a lot of them haven't been used yet, but they might hold a key for climate change. And what's different about apples is you go to the store and you buy grapes, and it might be PLU 4022, but it's not a name. So we're lucky to have a name, but we're also stymied sometimes because there's over 14,000 documented names, and I can't use one if it's been used before. So I pray that during my time, we won't get to the day where I'm doing a pharmacist kind of name with letters interspersed, but at least there won't be, could be a danger of diabetes and heart disease. So I think we're going to be okay. So this shows you there's 17 chromosomes. And so if I find something on chromosome 10, I also have to look at 15 because they're very similar. And where you see the TE density, that means where jumping genes have been noted. And the methylation is where genes have been turned on or turned off. And we'll talk about that a little bit later, but On Snapdragon, you'll see, or Honeycrisp, you'll see areas where the fruit is red. The gene for red in anthocyanin is turned on. The areas that are pale, the color for has been turned off. And then I found this was fascinating because we always think that when you have offspring, you're getting recombination, but largely a good mixture. And this is a Honeycrisp pedigree. Honeycrisp is very, very important in the industry, but you can see like where there's a large orange bar, that came from one ancestor. So there are mechanisms that prevented it from crossing over. So this is incredibly amazing technology that we have now, where it's 23andMe, but on a really global stage. And actually, it was 23andMe because they were able to use this to determine the parentage of Honeycrisp, which was wrong. And I laugh because you can get the pollen parent wrong because a bee can come, but when you get the female parent wrong, you've made a pretty big mistake because that's a tree that you should have been able to get the fruit from. So Honeycrisp is wonderful. My friend Jim Luby gets mad because he's only gotten mad when he saw the ad for Snapdragon that said, "Move over, honey, there's a new crisp in town." He didn't smile at all. But Honeycrisp, only about 40% of the fruit that growers produce get to market because it'll have soft scald or bitter pit on the bottom. That's a calcium disorder. It gets storage deficiencies on top. The leaves don't fully produce sugars. And so I wanted to take Honeycrisp and cross it by something that would mediate those disorders. And Snapdragon actually did that. I crossed it by 752, which is very similar to Jonagold, which our program released. And then once we have an individual we like, and in this case, it was the first tree in a row, which your stomach's fresh. And I always say, we can't select this because we haven't eaten 100 apples yet. We went back to it, knew it was good, and we'll take a bud from that and propagate it and make hundreds of copies. So just like the McIntosh apples that you eat from the grocery store, all of those are derived from budwood from that original 1700 McIntosh tree. So it's just like a giant Xerox machine that we're able to do with clonal propagation. So when we decided to do a club variety, which a club variety is different, where Honeycrisp right now, the patent expired a couple of years ago. And most people think that Honeycrisp is a relatively new variety. But that's why club varieties, where you pick certain growers, you say you need to use a certain amount for marketing, and you set quality standards, are important. But we wanted to do a club variety different than other places had done. So for example, for Sweet Tango, they gave it to one grower in Minnesota, and then lawsuits broke out like crazy. So we allowed every grower in New York State to have a chance to join the club. And about 151 of the 674 growers joined. And a lot of these people joined without ever having tried the fruit. They were confident in what we had done and where we were testing materials. So at the signing, we had the Commissioner of Ag, and he said, "I am so glad you did this, because if I had to tell the governor that you sold an apple to Michigan, it was going to be a long walk back to my office." So this was nice. We had no one complaining about, not having the opportunity. And, some people said, "I didn't sign on and I'm kicking myself now." And of course, being as diplomatic as I am, I said, "You should be." But they did. Okay, so seeing your varieties in the grocery store, I mean, I can't even tell you. So when it first came out, I said to my husband, "What's the reaction?" And he said, "People are lining up for miles." So not necessarily the case, but we do get fan mail, and to hear people, just there's a little boy that when Snapdragon runs out, he said to his mother, "Why can't people share?" It was just like, oh. So this made my day though. So the Buffalo Bills have named Snapdragon their apple for 3 years. And they're going to give away apples at the stadium 2 games in a row. And I have to tell you, I'm a big Bills fan. Football's always been big in my family. I still remember at the reception, you asked where the football game was. And so they were going to give out apples, and instantly I thought, that could be a bad idea because they can be like missiles. So instead, they decided to give out 2 or 3 slices and those convenient bags, which will be less problematic. But this has been wonderful. The Bills only have Snapdragon apples, and they're going to have commercials. They're going to have it in the concession stand. And when I announced this, my daughter said, "What do we get?" And I said, "Nothing." She was like, "Nothing?" But we get bragging rights, which is wonderful. So it's rewarding to see your creation being enjoyed. So Snapdragon has had, Ruby Frost is doing well, but it appeals to a person that likes a tartar apple. And it's really good for the sliced market trade. But Snapdragon really has had a meteoric rise. And so I'm always the crazy apple lady taking pictures at the store to see, what is competing. But a German consortium of farm markets has decided to embrace this variety, and they got the licensing rights across several different countries. It's also being tested in Australia and New Zealand, and it doesn't get better than that. So our growers have benefited, and now other growers, and we may have reciprocal deals. So in case you don't know, there are so many apples being created, and there's some really inappropriate apple ranking sites, but it's so funny because they'll say, well, this was bred in nature as opposed to a basement laboratory. I was like, what do they think we do in that basement laboratory? So the top variety now is Honeycrisp, Gala, Fuji, Granny Smith. Is that all other apples? Yeah. Delicious, Pink Lady, Golden Delicious, and Macintosh. Macintosh is still New York's #1 variety, but it's fading fast because it doesn't store as well. But these are going to change. But Red Delicious is the apple everybody likes to hate, but it went down and now it's going up again. And they tease that it's the apple of captive audiences. So schools, conference centers. So there's still varieties, and my variety is also in the all other category. So people say they're confused by too many apples. And do you feel confused when you go to the store about what you're going to buy? So I think you're getting confused because that's like a food fight broke out at Wegmans. So Snapdragon is in the box, but it says Fuji's behind it, and it's not. Sugar Bee is not Granny Smith, but that's what's in the Sugar Bee box. And so I went to Wegmans, and Empire and Ruby Frost were together. And I said, you can't have these together. It's hard to tell them apart. And the kid said, oh, anybody can tell them apart. I said, I made this apple. And that's when they're like, break, I'm going on break. Some problems we're seeing with, and you may see this when you go to the store, some problems we're seeing with climate change is lenticels are the tiny dots on apples that allow an apple to breathe, to exchange gases. But with climate change, it's a key area for pathogens and diseases to get in, and also for there to be collapse. And the sad part is sometimes they'll put apples in storage, they look perfectly good, and then they pull it out and it has these lenticel pits or burns. Every variety seems to be susceptible. This is going to be an area that we all have to study. So diversity, this is, I have some orchards where we just have bizarre things. And growers who have visited said, you are either genius or crazy. So I'll let you be the judge. A normal apple leaf looks like the small leaf on the rock. The bigger leaf is from a columnar tree, which is a single gene mutation. plus other genes that make leaves that almost look like tobacco. I mean, they're just huge. The little leaf is actually, the leaves go up and down the stem, which I've never seen any apple tree do that. It looks like a rosemary bush. And what's interesting about it is it has certain compounds that are really beneficial for health. The lobed leaves on the mat, I have summer scholars that come for 8 weeks, and they come in the summer, so apples aren't ripe yet. So I developed populations that they could study based on leaf morphology or flowers, and I share the data. And it's wonderful, but when you get red leaves, they usually fade. So you see the red leaf on top and green on the bottom. Well, under my caption, that beet red leaf is where we're able to get several promoters beefing up the color, and it stays red all the time. And so we'll talk about this a little bit later. So flowers, usually apples are single petals and white. This is the diversity that we have for flower color. We have one that's lilac. We have one that looks like a gardenia. I was an ornamental breeder at University of Connecticut at my start. So this is, my technician and I love this because you go out and we have drone footage and people come to take photographs and, zoom home to show their mom. So it's really something that's special. We're learning information, but we're also bringing some joy to other researchers. So look at the diversity. Look at that tree, the different flowers, a flower that looks like a daffodil, double whites, and all of this could be targeted for different traits. So we would love apple trees that set one center blossom, because we spend a lot of money thinning out fruit. And I don't know if any of you have home orchards, but the problem is nobody likes to thin fruit because it's like, but I might hurt the tree. I see a sideways glance there. So you really have to thin, because the seeds, if you have too many apples, the seeds send a signal that say no fruit next year. And you get into this dangerous habit. So if we could study some of these that set only fruit at that king blossom, that would be tremendous. So I like to do this just because I have great respect for the woman scientist, Barbara McClintock, but she was really instrumental in looking at corn that had different pigmentations, different patterns. And she gave the hypothesis that there's something called jumping genes, which can, within the DNA, insert and excise. And if they come in near a color gene for the pathway for anthocyanin, it may either enhance it or decrease it. And she was ridiculed. And then years later, she got a Nobel Prize for her work. So if you have a chance, the book Feeling for the Organism was wonderful. And then there's a review in Cell that one of her students explained that he brought flowers for her, and he was worried that it might be deemed inappropriate. And she said, "Oh, variegation." And she took out a scalpel and cut the carnations and started explaining where the different changes occur. So it's worth the read. So this is the genetic diversity we see for plant form. So that I call the tree that's like a bush, the cousin it tree from Addams Family. I don't know why, but it resonates with people. We have weeping, we have segregation for branching or not. We have different heights. The tree that looks like bicycle wheels is not pruned. It's an auxin mutation, and it just grows like that. So the reason that this is interesting is we could create the perfect tree for robotic harvests or for individual harvests. And so this allows us to study in detail what causes the branches to form like that, what hormones are involved. So I thought this would be interesting because when I travel, a lot of people don't realize the evolution of our orchard systems. And you're close enough to Cornell orchards and other orchards, but In the past, we had old freestanding trees. That picture was taken 2 years ago at a Connecticut orchard. So they still had trees on large rootstocks. And what we used to say is they're growing firewood rather than fruit, because the amount of wood that they're producing is more than the apples that are produced. And it's with labor, that's a concern. So now we have trees that look like they're, just stakes. We have dwarfing rootstocks. We want to harvest the sun because the more light exposure you have, the better the fruit. But it's also much more expensive. But we have that trellis system platform where pruners can be on the platform for harvest and for pruning, so a lot less back injury. And a lot of the farms have this technology. We have fruiting walls, and then we're going to have grants for mechanical harvested of apple. And I'm still skeptical because the first one was like a regular vacuum hose. And when I had toddlers, all I could think of is, if you can suck up baby socks, you can suck up leaves, branches, and debris. I didn't know how that would be done. Now they have grippers that can harvest it, but the human hand can harvest it so quickly that we have to have evolution of that form. Shrubbery robotics are there, and a colleague of mine with weeds has an electric zapper that actually can recognize weeds and zap it. But apple has a little bit steeper learning curve to go. But as I said, we have rootstock breeding, where the reason we're able to change the density and management is that we used to have the biggest trees, and that's why you had big. So there's the scion, my part, and the rootstock, which Jim Cummins and Gennaro Fazio work on. But now they have trees that if you put snapdragon on the smallest rootstock, you're going to get probably too small a tree. And then as you go up those classes, you're getting bigger and bigger trees. So you can actually custom pick a rootstock for a variety. And growers are spending a lot of time doing that to great effect in terms of profitability, and also bringing in, you don't have to wait a long time for trees to fruit. They can fruit in the second or third leaf. And so profitability is higher. So when I talked about chimeras, we have so many chimeras in apples that we actually have a competition who can outdo each other. And if you go to a gala bin in the grocery store, you can find some too, and some of the peppers, but I lost. That's a four-way chimera, the big apple. The apple with russet, that is half russet or quarter russet and quarter not. My student used to determine that the russeted part has so much more of a nutritious compound than the non-russeted part. So these sports can help us look at very interesting questions. We have a chimera of the leaf, and then the columnar habit, the spur habit. So apples used to grow on long shoots, and now they grow in short spurs. All of those are due to transposons and insertions. And I have a student that's working on cannabis, and he was talking about a gene, and he goes, well, this gene doesn't work. Well, guess what? There was an insertion. So it happens much more than you think. The variegated plants that you grow are likely due to a chimera. And so if you've ever planted a variegated boxwood or any other plant, and then you see it revert to green, it's because the green is more photosynthetically active. And so you're spending a lot of money on a variegated plant, and then you turn around and say, I could have saved some money. But this is where it gets complex. We used to think that the original Red Delicious was a poorly colored apple with great quality. And when I show the, let's see, when you see the 4 different headings, it's a first mutation for color, and then that tree mutated again, that particular variety mutated again for color. And we used to think, it's just a simple gene, some more anthocyanins and less volatile. So that's why Delicious today is nice and red, but it doesn't have the volatiles that contribute to quality. But we're doing this with all our varieties, with Fuji and Macintosh. We're always chasing color. But you can see that in some cases, there's 542 differentially expressed genes. So it's very complex. And so when I see a researcher that's saying, oh, we just have to make this simple genetic change, it's like, not quite so simple, but we have the technology to get at it. We have plenty of diversity. We have the USDA collection in Geneva. They have an open house every Saturday. That might be a good program idea. And then what I tell all my students is understanding variation is key. I know that a lot of you understand this, that if you don't understand the developmental stage that you're at, and I'm very fortunate that I've always had people like, I don't know, I'm going to go, a lot of physiologists, post-harvest physiologists that have been able to help me understand what is the optimum stage. Dave Blamke came to my program and he was showing me how to store fruit. And Dave and all the, everybody that's a pathologist loves to see disease. Entomologist loves to see insects. And Dave is a post-harvest person. We took out material and he goes, oh, Susan, I've never seen so many disorders. And I'm like, but he was excited. We got past that stage. But this is what shows you like how incredibly complex it is. So we know nowadays we needed cold temperatures because cold temperatures help apple color to develop. And you also see with the sticker that sunlight is important to coloration. But then look at all the pathways. So there's promoters that turn on color, and then there's things that interchange with that. So yes, we can make progress, but you have to understand all the steps or know somebody that does. So we are going to start seeing red fleshed apples. Now I'm staying as a breeder because I once said that rocket apples that they sell in plastic containers would never go anywhere. And then they had a major multi-million-dollar investment. So it's like, okay. So I don't think red apples are going to make it because you might buy one as a conversation starter, but I don't know that you're going to pay a premium when you could just eat 2 apples if you want to be healthier. And so what I did is I had a student class come and I asked for their permission to tape them and take a picture of them after eating a really red fleshed apple. I love this picture. I may frame it because their reaction was so spot on. And they were like, this isn't fair. And I was like, this is what breeders do. You have to try them to see. So red color can be tied to one gene from the center of origin, but it has a lot of acidity and a lot of astringency. So you have to try to blend it out. What it does make is an excellent rosé hard cider. And so that's where my money is. But again, I've been wrong. So this was just an example of how a generous Cornell professor helped me, but also, serendipity and being prepared. I had a student that wanted to look at pale green lethal. That's when that little plant, the seedling comes up, it turns yellow, it dies. Catchy name. So he did all sorts of genetic studies and he said, there's a problem with phyloquinone or vitamin K. It's like, okay. So I'm in my office and I get a call from Dr. Parker. And I always Google people before I talk to them. Not that I think that they're going to be strange, but because I want to be prepared for what their expertise is. Well, guess what Dr. Parker's expertise was? Chemistry, and specifically vitamin K. So I said, could you help us on this project? And he said, sure. So lo and behold, he found out that the pale green lethal was not producing vitamin K, but if you sprayed it, you could rescue the plant, but you couldn't make it as tall as the other. So then we started spraying and I had my poor student spray and spray. And finally he said, can I stop? And sure enough, you can see where he stopped is the blue bar. And then once exogenous vitamin K isn't added, it goes downhill. But the beauty of this is that lethal is linked to a lot of important traits like fruit acidity, storage, bitter pit, And so now we know that we have to be really careful in parental selection. So it was a really breakthrough study. And then I had two Bens in my lab at the same time, which is not recommended. So it got confusing. But Ben and Sully, and when I saw this picture of Sully, I said, what did you tell your student about me? Because he does look petrified. But Ben did a really nice study of looking at the germplasm repository for 3 compounds, fluridazine, trilobitine, and sabolden. It was initial research from like 1960s. And it separated Malus species, but they found that these compounds can actually be used in diabetes research. Glucose metabolism, they're actually being used for some drugs. He was able to determine their inheritance, where they're located, and we've had major companies come in because they're also bitter blockers. So I had wanted, and I didn't get a chance to bring to this group, there's something called miracle fruit, and you can take one of the berries and then you can eat a lemon without the astringency. That's kind of what they are. If they're added to beverages, it can block the bitterness and enhance the sugar. So my next release, and why I'm going to be still staying here a while, as Lisa said, 37 years. I had a breeder in his mid-80s, and he called me and he said, so when are you giving this up? He didn't say he was. So I replied, are you my husband's friend? So I have to release this russet. It's the most amazing apple that I've ever had. I showed it to some marketers. They said they could sell millions of them. I'll give a warning, and I didn't have enough, even if my shoulder was good to bring, but it's like an old time russet, but it doesn't get the black spots. Very crisp, juicy, wonderful, nectary, but it has a hint of anise. And 50% of you will like it, and 50% of you will hate it. And I had one woman that tried it in my office, and she was actually like scraping her tongue. It's like, okay, I get it. You don't like it. And then the other release is an ornamental crabapple with the columnar gene and double flowers. And it's absolutely breathtaking. So that would be for landscape. So Larry Smart in our program went out and said, I want 12 of those. So that's kind of an homage to my mom. Variety that you may have tried at Cornell Orchards, it was either selection A or B. Sadly, they changed the sign, so I couldn't always tell which, but it's a modified russet, really high sugar, high acid. And we named it because the cider makers were making hard cider out of it. So there is a Snapdragon's hard cider from Beacon Skiff and also Angry Orchards, but there's also a Firecracker hard cider. So the challenges that we face in the future, invasives are going to be huge. I mean, you've all probably starting to see marmorated stink bug. My husband said he went skiing and put down his ski goggles and then screamed like a little girl because there's a stink bug right there and he wasn't expecting it. They like yellow and you don't want to squash them. Ambrosia beetle is really cool because it has a sac on its back. It goes into the tree. It dumps the sac of fungi that then grows, and that's the ambrosia. It feeds its young with that. It makes injured trees smell like alcohol. So we're using alcohol baited traps to monitor them, but it's a devastating pest. And then the one that you hear about from NPR and everything else is spotted lanternfly. It devours everything. There's not going to be genetic mechanisms for breeding for this, but citizen science to make sure that you're aware and even if you love bugs, kill them. And then climate change. We had the first time that we lost a crop completely was 2012. We had a lot of frost right before bloom. We also had hail that was devastating. And so we are seeing many more occasions of this snow on blossoms. A lot of orchards are using hail nets. Washington and Oregon are going to be suffering from drought and sunburn. In Washington, they had a heat dome that I think the temperature was 100 and it was high. It was in the low to mid hundreds. And the fruit can be like 30 to 40 degrees higher than the ambient temperature. So a lot of sunburn, and they know that that's going to happen again. We rarely see sunburn, one benefit to a gloomy area sometimes, but we are seeing more of it. We're seeing rots that used to be only in the south. But I did love the meme of, meanwhile in New York, when we were having all our winter, our water challenges. So we are looking at it, and we are doing test crosses to see what's adapted, but it's hard. And then COVID. I mean, we all know COVID, but apples are just as bad. So there's all these viruses that are mutating and infecting apple. And we have a virologist that's doing his job. He's a Clean Plant Network, but he studies finding new viruses. But if he finds a new virus, even if he can't tell me it causes problems, I can't send my material to other places. And there's just a ton of them. So then, if any of you worked on Arabidopsis, there's just an article the other day that they found that a lot of the Arabidopsis stock, which is like, the racehorse of plant genetics, were infected with virus, and that's going to impact your results. So we're still battling this. We are trying to get clean plants propagated, but that's why, I once got budwood in the mail, and my husband said, "Oh, look, it says children's toy." And I picked it up and said, "This is budwood." And somebody was meaning well, but that's how stuff gets introduced. So, I gave it to the state inspector, and he said it was hot as anything. So, try not to share things outside the region. I'm excited about the other opportunities we have. I wish I could stay in this job forever, but eventually I'm going to have to quit and then join CAPE. So I hope you'll have me. There's many more in the pipeline. That spotted apple, I think it's really good quality and really unique, all the lenticels. But I asked a audience of like 100 to 200 growers who found it attractive. One person, it's like, this is a tough crowd, because I figure you could be distinctive in that. So we'll have more releases for both the general public and for growers. We'll have stuff for hard cider, and who knows what new opportunities there may be. So I thank you very much for inviting me to speak today. I'm glad to answer any questions that you have. I've been bad because I haven't really looked at the chat, but I've learned that my skills for multitasking are not that good.